Thanks. It's a pleasure to be here. Especially, I've got that coveted spot between you and lunch, and so I will try to get through this. Some of the things that I'm going to say have already been presented this morning. Kurt and I work together on some things, and what I want to give my acknowledgements up front, because I'll get to the end of the talk and not put these people or acknowledge these folks. Paul Carter, Columbia County Extension, who we work with. Sam Jayasingh, who's a grad student in my program. Kurt, Hai Ying are both collaborators on our work. And then Steve Van Vliet, Whitman County Extension as well. So Kurt presented some of this data. This came out of the University of Idaho about the time that I started working here. And this was Bob Mahler's soil survey, or a survey of soil lab test data, that first really drew attention to some of the soil acidity problems that were beginning to develop. And basically what he had done is looked at soil test records for the previous 30 years from a variety of different labs. And it shows here 1960, 72, and 80. And this speckled area is less than 15% of the acres below pH 6. And then as you go up, this crosshatch is 15 to 45, little denser, over 50%, and then over 65%. And basically, this is the story that you see that you've heard about already today. This problem that we're dealing with today started a long time ago, and the culprit really is, as you've heard already, mainly nitrogen, ammonium-based nitrogen fertilizers. So why do we care about this? Obviously, again, you've heard some of this crop productivity. Again, data from Bob Mahler's program, and you have lentils, barley, winter wheat, and peas. And these show a stable yield until you reach a certain pH, and then it begins to decline. And Mahler called that the minimum acceptable pH for crop productivity. And this was done several studies, multiple varieties, multiple locations. And so these are kind of the data that we're looking at, that we're calling attention to this problem of soil acidity. This is another form of that data for some other crops. Alfalfa here, basically what you see is this is percent of maximum yield. So we have 100% here at the top. We have pH across the bottom. And you'll see here that at the lowest pH, this is actually a winter wheat that is more tolerant of aluminum, or excuse me, of low pH. This happens to be around pH 5.2, where you start to see pH going down, which is also a point where you start to see aluminum coming up. Okay. So what are the reasons for reduced productivity in these low pH soils? One can be direct toxicity from various elements in the soil, lack of cationic nutrients, loss in bases, low phosphorus availability, altered soil microbiome. Tim Pollitt's talked about that this morning. One especially is toxicity to the rhizobia that form galls, nodules. And then my job today is to talk about plant diseases and the impact of pH, soil pH on plant diseases. So here's an example. You've seen something similar to this. This one actually comes out of Australia. The Australians have dealt with acid soils in cereal-based cropping systems for a lot longer than we have. And basically what you see are this is a subterranean clover. That on the left has a nice, robust root system with a lot of nodules, which we can't see in this photograph, compared on the right with the same crop grown in acidic soil. Root system is limited, and there's limited nodulation, and so there's going to be reduced productivity. This gets to that change in the soil microbiome that Tim was talking about this morning. So when we talk about diseases, soil scientists always talk about the nitrogen cycle. Plant pathologists always talk about the disease triangle. And so the three things that we have to have for that disease to occur is an environment, a plant, and a pathogen. And sometimes we think about pathogens as being only those living kinds of biotic organisms, and a lot of times they are, but sometimes they're also the abiotic environment. And when these things come together, then that's when we see a plant disease developing. Okay, so we think about environment, the plant, and the pathogen. And in the environment, things like soil pH, temperature and moisture are very important, but other things like pH. And with the plant, we can have resistance and susceptibility. And you've already heard about breeding for aluminum tolerance in plants, another kind of breeding. And in the pathogen, some other attributes that we won't really deal with today. So in the Pacific Northwest, inland Northwest, especially in our wheat-based cropping systems, we have a lot of different diseases that can potentially develop on our crops. Where those diseases develop, how severe they are, relate a lot to temperature and moisture. Okay, some of these diseases are going to be found more in the higher precipitation areas, some more in the lower precipitation areas. But then we also have soil pH that is impinging on these diseases too and can have an impact. And I'm going to talk really about all of these diseases a little bit, some more today. Except for stripe rust, the snow molds, and soil-borne wheat mosaic. We really don't have any data to show that these diseases are responding to soil pH one way or the other. And I'll give you the punchline right now. We'll share some data, but Decay and Cephalosporium stripe are the two diseases out of this group that respond more to soil pH than any others, and they respond differently. Cephalosporium stripe is going to be worse at the acid end, and Decay is going to be worse at the alkaline end of our normal sort of pH range. Okay, so we'll start with Fusarium crown rot. This is, again, caused by a soil-borne fungus. Some of the symptoms of this disease, you'll see these dead standing stems. This disease is often associated with water stress, or at least you see the symptoms become more apparent with water stress, and it results from the fact that the fungus has infected the roots and the crown. It chokes off the water supply, and when the plants come under water stress, then they die, and it can have a significant impact on yield. Here's another example, Fusarium crown rot. You see this kind of chocolate brown discoloration on stem bases and subcrown internodes. Dick Smiley at Pendleton with Oregon State did some study in their long-term plots, and he published this work back in 1996. And basically, they looked at pH changes due to nitrogen fertilization and found that soil pH was inversely proportional to the rate of applied nitrogen. What that means is that more nitrogen, lower pH. And also organic nitrogen and carbon. And that disease was also inversely proportional to soil pH. So if we look at these figures, this is crown rot, and this is based percent whiteheads. And so we have bulk soil pH, so we range from 4.3 to 5.5. And what you see is that there's more disease at the low end of this run than at the higher end. Okay, this is over about 1 unit of pH change. And he also looked at organic nitrogen in the soil and found that disease was associated here with higher organic nitrogen in the soil. So Kurt Schroeder shared this slide with me, and he also looked at pH relative to Fusarium crown rot. And his data shows something a little bit different. These were in inoculated plots, pH from 4 to 7, and then a disease rating on the left here. And what you see is that the low inoculum, no inoculum rate, there's not much of a response. Maybe a slight increase here at the mid pHs. With a low rate of inoculum, it's a little bit more apparent, and with a high rate of inoculum, it's even more apparent. But here again, we're looking more at this range of pH 5, the highest here at pH 6. So there's not what I would call a real strong response here to pH. something of a response and a little bit contradictory between Kurt's results and Dick Smiley's results. Rhizoctonia root rot, another soil-borne fungal disease typified by these spear tips on the crown roots. The fungus gets in, it rots the cortical tissue on the roots break, and then you see these kind of spear tips. Also called bare patch because in fields you can see erratic stands. Sometimes the stands are simply thin. Sometimes you actually have bare patches where there are no plants present. Okay, again, Kurt's data looking same sort of situation with pH and disease rating. With no added inoculum, there's really no response at all. It's pretty flat. As you increase the inoculum rate, you see a slight increase in disease here in these middle ranges of pH, but not what I would call a real strong response. Okay, something, but not a real strong response. Okay, Pythium root rot. This is another soil-borne fungus-like organism. It likes wet soils. It has a swimming spore stage. Typically, it will infect root hairs. You can see a healthy root here on the left, an infected root on the right. This one has root hairs. This one doesn't. There's some discoloration here. When you look at it in the field, this is a field plot from many years ago, a metalaxyl treatment, seed treatment on the right, and a check on the left. And there are differences in stand and in the rows filling in and so forth. When we look at the response of pH, this is some work that was done by a graduate student of Jim Cook's many years ago. pH across the bottom here ranging from 4 to 8, and this is the incidence of embryo infection. And basically what they did was to take two different soils that had different starting pHs. This Larkin silt loam here on the top had a pH in the beginning of 4.6. This Padigo silt loam started at 7.6. They adjusted the pH of those soils. They put it into pots, and then they watered those pots, planted seeds into them, and then pulled the seeds out after 48 hours, put them onto a growth medium to determine whether they were infected or not with the Pythium. And basically what you see here in both cases is that at the low end, in this one case, there's actually low infection, and low infection at the higher end, 7.5, but the greatest response is kind of here in the middle, but it's pretty flat from around 6.8 over here to about 4.8 in this soil. In this soil, you see a little bit more of a decrease, but again, pretty flat. There was infection here, though, down to pH 5. They did treat these soils also in some of the treatments. They added an antibiotic, which would be inhibitory to certain bacteria in the soil, and they found that there was greater infection at a higher pH when they treated with an antibiotic. And then they concluded on this, that the microbial environment in the soil around those seeds was having an impact on infection, and that by eliminating some components of that they also changed the pH response. So this gets back again to some of the things that Tim was suggesting earlier, that the microorganisms in the soil can have an impact on how that plant responds to that pH environment. Okay, this is not wheat. Weidong Chen shared this with me. This is for chickpeas. They have found some of these spots in fields where Pythium seed decay was more of a problem in chickpeas than other spots. This is from a few years ago, and they went into these spots and they dug up some of the seeds that either did not emerge, or some of the plants that were not growing very well. And they looked at several different locations for this and characterized the isolates of Pythium that they got out, and they found that, lo and behold, that some of them were resistant to the metalaxyl fungicide. Okay, so that we kind of knew was developing. Inside the spots, there was a greater proportion of resistant isolates than outside the spots. But interestingly, when they looked in these spots, They found differences in soil moisture content, which wasn't too surprising. If you don't have plants growing, there's nothing there to use the moisture, so you have higher moisture content. But they also found a difference in pH, that inside these spots, the pH was 1 to 2/10 of a unit lower than outside the spots. Not sure what that means. I'm not sure that they have an interpretation yet for that, but there is this association with a slightly lower pH being more favorable to this seed decay problem. Okay, Eyespot or Strawbreaker foot rot, another soil-borne fungus disease. The fungus infects the stem bases, causes these elliptical kind of lesions on the stem. Smiley, again going back to the study in their long-term plots at Pendleton, found that the incidence of Eyespot was proportional to soil pH, inversely proportional. Okay, so as soil pH decreased, the incidence of disease increased. This is the only report in the literature of this response, and there was no data really provided. And the size of the response is not known. In full disclosure, this is the figure that I showed you earlier for Fusarium crown rot, because no data were provided. And so I have to say that for Eyespot, the jury is still out on whether there's a pH response or not. I've worked with this disease for over 30 years, and in our plots where we are working with soil pH, we have not seen a difference in Eyespot, but we haven't really tried to quantify it carefully. So that's why I say the jury is still out. Okay, take-all. Take-all, another soil-borne fungus disease. It can cause whiteheads in standing wheat. It is a root-rotting fungus. It can cause discoloration of the roots. You see these, what we call runner hyphae, on the surface of the roots, and it can also cause this discoloration on stem bases. It can be a very severe disease when it occurs. It tends to occur in wetter areas. In our part of the world, we tend to see this more in irrigated systems, especially when you have short rotations between cereal crops. Okay. This disease, again, this goes back Dick Smiley when he was a student with Jim Cook back in the 70s. They looked at the incidence of take-all and soil pH, and that they found that severity was related to pH and form of nitrogen. The disease increases with increasing pH, and this is what we've known about take-all for a long time. The application of ammonium-based nitrogen resulted in less disease than the nitrate form of nitrogen. And the reason for that is that when plants take up ammonia, the rhizosphere becomes acidified, and that's less favorable to the fungus. And these data try to show that here. This is bulk soil pH across the bottom. In this figure from 4 to 9, a disease rating. Two different locations, and what you see here, they applied different forms of nitrogen in these plots, and you see here at the Ritzville and Puyallup locations both, the lowest disease is when pH is below 6. As the pH increases above 6 here, the incidence of disease goes up as well. In this figure on the bottom, this shows rhizosphere pH across the bottom from 4 to 9, the disease rating on the left. Two different forms of nitrogen, calcium nitrate, which is neutral in terms of pH, and ammonium sulfate, which is acidifying. Again, what you see here is an increase in disease with increasing pH. Form of nitrogen here, there was less disease when you had the ammonium sulfate applied compared with the calcium nitrate. Okay. And then Cephalosporium stripe. This is a soil-borne fungal pathogen. It is a vascular wilt type disease. So the fungus infects through the roots. It colonizes the plant from through the water conducting tissue, the xylem. And like many vascular wilt diseases, this disease is favored by acid soils. It can be quite severe when it occurs at a high level or a susceptible variety, causes these dead standing stems, and it's about a 1-for-1. If you have 1% infected stems, you have about a 1% yield loss. We know, going back a long ways based on the scientific literature, that this pathogen likes acid conditions. In 1964, there was some work that came out of WSU that this fungus produces an antibiotic. There's more produced at lower pH. This is in culture. Another study in 1968, survival of the fungus was enhanced at low pH. And then in 1985 was really the first indication that we got that low soil pH favors disease development. This came out of Kansas State University. And this was a study that was done. Soil pH is shown on the left, and then these are cropping seasons following the initial start of this experiment. And the way this works, they looked at both Cephalosporium and Decay, and they had 3 treatments. A control treatment here in the dark red. They added sulfur at 430 pounds to increase pH, or excuse me, to acidify the soil, and then they added lime at 5,000 pounds per acre to raise pH. These are the starting pHs here. In the summer of 1981, all of the treatments were at 4.8. By the fall, the treatment where they applied the lime had increased to 6.3. The other 2 stayed the same. The following spring, the control and sulfur were about the same at 4.6, 4.7, but the lime treatment increased, and this continued through the experiment. So really the only response to pH was in the liming treatment. Okay, when they came in then and looked at disease, this is percent disease on the left from 0 to 60, the 3 treatments, and then they looked at Cephalosporium stripe in 1982 and 1983, Ceph stripe and Decay. So these first 2 on the left here are Cephalosporium. These 2 bars on the right are Decay. So this is 1 year after application of the lime and the sulfur. Again, you see the pH here was 4.8 in the control treatments, 4.8, 4.6. About 28 and 20% Cephalosporium in the control, about 10 and 7% Decay in the control. In the sulfur treatment, there was about 33 and 18 for the Cephalosporium, and about 6 and 11 for the Decay. Not much change in the Decay, a little bit more Cephalosporium. In the lime treatment, Cephalosporium was at 15 and just less than 1%, but the Decay increased dramatically here, 50 and 56%. So we get just the opposite kind of response here to liming. Cephalosporium decreases compared to the no treatment control, and the Decay increases dramatically. Okay. So a graduate student that worked with my predecessor, Bill Briel, Connie Love and Bill Briel published this work, and this is right after I got started here. And basically what they did was to collect field soil and then adjust the pH. And at the time, the field soil pH that they collected was around 5.7. And so they adjusted it down to 4.5 using sulfuric acid, and they adjusted the pH up to 6.5 and 7.5. They took that soil and they put it into these what we call microplots, which were basically boxes that were 3 feet by 3 feet by about 10 inches deep. They planted a susceptible variety of wheat into it, 6 rows, and they inoculated 3 rows and left 3 rows uninoculated. And I'm showing you just the pH 4.5 treatment and the pH 7.5 treatment, and it's clear at pH 4.5 which are the 3 inoculated rows. You see the stunting and the whiteheads here at pH 4.5. But at pH 7.5, if I didn't have the signs up there to tell us which were which, we wouldn't be able to pick them out. And that's the kind of response that we're seeing to pH with this disease. Okay, we followed up on that work, and limed field soil to these same sorts of levels. We had target pHs of 4.5 to 7.5 at 1 unit intervals. And we looked at this over a couple years in the field. This was pH of our different treatments over time. And so you can see we had, at the end of our time here, we had pHs that were down around 4 all the way up to about 6.8 or so. And when we looked at disease, this is percent infected stems, soil pH across the bottom. 2 different years we saw this decrease in disease, and we saw a corresponding increase in yield that mirrored that increase in disease. Okay, so the bottom line here is that liming controlled Cephalosporium stripe, and it resulted in a yield increase. Okay, so I want to move on to what we would consider an abiotic kind of a disease, and this is aluminum toxicity. Borrowed again from Kurt, shared this with me. You saw this photo, well, you didn't see this photo earlier, you saw the next one. This is this variety trial near Rockford, 2 varieties, Scarlet and Alley Cat. Pull up the plants, you see the root systems are quite different on these 2 varieties grown in the same soil, right, adjacent to one another. And this is the result of low pH and aluminum toxicity. Okay. Again, turning to some of the work that's been done in Australia with aluminum toxicity. On the left here, I put this in because it's a pretty dramatic photo. This is a tolerant, a wheat variety that has aluminum tolerance, and this is a wheat variety that is sensitive. And these are 4-day-old root tips that were grown in 5 micromolar aluminum chloride. And within 4 days, you can see the kind of damage that is occurring to those roots. This photo up here, it's a little harder to see, but you have tolerant and sensitive varieties that were grown again in, this was 50 millimolar aluminum chloride, and then stained with this stain hematoxylin, which binds to aluminum. And what you see is that in the aluminum sensitive variety grown in a solution with aluminum, that there's a significant amount of aluminum that has been incorporated into that root. And so at a very young age, aluminum starts to do damage to these plants. Okay, here again, another illustration of the effect of aluminum. Plants on the left were grown in soil pH 5.1 with aluminum less than 2 parts per million. Plants on the right, soil pH 4 with aluminum about 15 parts per million. And this goes back to what Kurt said, is once you get above 10 parts per million or so of aluminum, you start to get past that threshold where we begin to see damage. And these are 11-day-old barley seedlings. Okay, so at a very young age, aluminum toxicity starts to become apparent. Okay, so you've seen illustrations similar to this today already, but again, just to follow up, pH across the bottom, aluminum concentration on the left axis here, and as aluminum concentration is relatively low until you get to around pH 4.8 or so, and then it starts to go up dramatically. And you know, this illustration on the right shows availability of some of the elements for plant growth, pH 4.5 to 7.5. You get down below that pH 5 mark, most of our important elements are starting to decrease in availability. Some of these minerals are increasing in availability, and of course we're concerned about things like manganese, iron too much, aluminum too much. Okay, so we've talked about liming, and Kurt did a great job of talking about some of the issues with liming, but liming to raise pH locally, part of the problem is few local sources of high quality limestone, plus the cost of transportation. equals limited use. Okay, it costs a lot to get the material here and to spread. The other option for managing aluminum toxicity is breeding for aluminum tolerance. And I'll just say now, my own personal belief is, and like the speakers before me, liming is the solution. Breeding is going to provide us with some short-term benefits, but in the long run, we need to deal with the root problem, no pun intended, the root problem, which is low soil pH. Okay, so with that, I include this list of varieties. There are varieties out there that have tolerance to acid soils. Kurt showed some of them. This is the list that I compiled from this publication by former grad student of Aaron Carter's. And so there are some options for now. But going forward, we need to, as I said, think about how we're going to manage the root problem. These photographs are from the breeding nursery up around Rockford, where Mike Pumphrey and Aaron Carter are working. I took these photos a couple years ago because we're working with soil out of this plot. Here you can see kind of a cross-section here across the rows. These tall plants here, that's triticale. And if you think back to Kurt's slide, triticale is the most aluminum-tolerant thing that we can grow in the region. Here on the lower right, these are a couple of lines that Aaron Carter selected as being tolerant, again, based on this single gene, this malate transporter. Aaron told me that in his breeding program, his definition of success is if the plant survives and produces one head, then that's good enough for a start. Okay. So one of the things that we've begun looking at is the alternatives, I shouldn't say alternatives to liming, but alternative products. And two of the things that we're looking at are paper mill fly ash and biochar. Paper mill fly ash is a byproduct of paper production. It's highly alkaline. It's mostly calcium oxide, about 60% or so. And it has a lime value of about 100 or so. We happen to be working with Inland Empire Paper Company. They approached us about this. They're generating about 20 tons of this material a day, and it's going to the landfill. Okay. Biochar is the result of a combustion of organic material in the absence of oxygen, and it has a low lime value, but it has the ability to add stable carbon to soil. And I'll just make one comment here about when we say biochar, it's kind of a generic term for any number of different organic materials that have been combusted in the absence of oxygen. The temperatures at which they're combusted, the amount of oxygen, all have an impact on the characteristics of that biochar when it's incorporated into soil. And one of the reasons that we were interested in biochar is not because we thought it was going to change pH, but because of its potential impact on soil microbial communities. And there's a lot of work that's been done, some in acid soils, on the ability of biochar to ameliorate some of the damage of acid soils, but not by changing pH, but by changing soil microbial communities. And so we wanted to look at the effect of these materials on Cephalosporium stripe, soil microbial populations, and basically growth and emergence of weed. And the study, and I don't have a lot of data to show you today, but we're working with two soils. This soil from that site that I showed you, this Rockford soil, pH 3.7, 230 parts per million available aluminum. And then a Pullman soil, pH 4.6, about 6 parts per million available aluminum. We have two different forms of fly ash, same composition, but they come out of the process at different points in the process. And then two different types of biochar that we're working on. This Amarone here is a forest product biochar, and the other one is an Ag Energy. It's basically a wheat straw based biochar. So we've mixed these materials at, all combinations of these materials. The biochar was about 2 tons per acre. The fly ash was about 2.3 tons per acre. We used a conventional liming product at about 4 tons per acre. Mixed these and then planted them into our what we call microplots. These are pots that hold about 10 kilograms of soil, and then we've measured pH about every month or so. And this work is actually from our last year's study. I waited too long. They're covered with snow this year, so I didn't get photos. But this shows like 1 rep of our microplot study. And you can see in here, I'll point out that these plots, this is the Rockford soil. It's more tan in color. The Palouse soil is darker in color. This is the check here, no lime, no biochar. And these other pots are the same soil where we've added the paper mill fly ash. Okay. And then we have the Palouse soil in here. We have the check right here. And then those where we've added the paper mill fly ash. And so if we drill in on these, here again the Rockford soil, untreated control, Rockford soil with fly ash, and then here's the Palouse soil and with and without fly ash. And you can see here we went from 3.7, pH 3.7 to pH 5.2, pH 4.6 to 5.9, 230 parts per million of aluminum, 12 parts per million of aluminum, 3.6 to 2.7. And then here is a summary of the soil pH, aluminum based on these various treatments. And across the bottom here, so soil 1 is the Rockford soil, soil 2 is Palouse. We have the 2 different forms of fly ash, no fly ash 1, fly ash 2, and then ag lime. And the same over here. And these bars represent the soil pH. So you can see here, no treatment, we're about 3.7. What I showed you, what I told you in that photograph, we're up over 5 here with the fly ash. The other form of fly ash was not reactive, came out of the process at a different point. And then the lime, we had an intermediate pH here in the Rockford soil. Most telling here, these 2 lines here show the available aluminum concentration. So we're over 200 here in the no check. We get into the fly ash and we're down very low. The ag lime also reduced aluminum, but not quite as dramatically. And then here in the Palouse soil, we had a very low level to start with. Okay, so the bottom line here, the conclusion is that this fly ash does provide an alternative to conventional ag lime. And we are expanding this into field scale trials. We're working at 2 sites. This site that you see here is near Rockford. It's not quite as severe as the site we were working with, the other Rockford site. This is about pH 4.4 and about 150 parts per million available aluminum. And we have the same experiment on the conservation farm. This just shows we checkerboarded our experiment out there, all possible combinations of fly ash, a couple forms of biochar, and an ag lime control. And then we incorporated it at both of these locations. So that was just this past July. At this location, the grower seeded winter wheat over the top of that. And at our location on the conservation farm, we actually did a small variety trial with 4 different varieties that ranged in their tolerance of acid soils. And the site at the conservation farm here in Pullman was inoculated with the Cephalosporium pathogen. This one not. We'll see what happens in that field. So I'll just leave you with this. There are some resources on soil acidification. And then be happy to take any questions. Yes?
The cost of the fly ash and biochar, how does that compare to the liming product?
Yeah, the short answer is on the fly ash, we don't know. Inland Empire Paper is interested in finding an outlet for this product. One of the problems right now is the form that product's in. It's a very flowery, light material. It's not very user friendly to spread. We've been talking with them about alternative formulations. If we could find, for example, a prill that could be spread with a spin spreader or drop spreader, it would make it a lot more user friendly. So I don't think we're anywhere near getting a pricing on that. Biochar, the price of biochar is all over the place. It's more expensive to apply, and for that reason, I didn't mention it, but in our field plots, I think we limited our applications to about 500 pounds per acre. The other thing I wanted to mention is that in our field plots, we've sort of been trying to think about how to address this problem of what value we should lime to. And so we've gone to using liming to a lime requirement of pH 5.7. And we did that based on Mahler's initial work, which showed that for most of our rotation crops, we still maintain 100% yield potential at pH 5.7. The other thing is that in some of these soils, if you're down below pH 4, you can't apply enough lime. If you apply the lime that you need all in 1 year, you're going to have other problems with putting that much lime on in a single year. So you may have to split applications. But like has been said already today, I think you're better off putting a higher application rate on initially and then looking at fine-tuning later, monitoring the pH and fine-tuning later. But we're in the 2 to 3 ton per acre range at both of our locations based on that pH 5.7 lime. Yes?
Fly ash is free. We're going to pay you to haul it out.
Right now it is. At the flowery form of it, you could probably get it for free.
And to solve the spreadability, we do this at IEP.
Okay.
IFG, I'm sorry. A garden hose and a T-jet nozzle, take it to 15% moisture. We pushed it to 20. You don't need it.
Really? With the fly ash?
Yeah. So number 12 T-jet nozzle and a garden hose in the bottom screw auger takes it to about 20% moisture. Stores, throws, hauls, pretty easy. They'll pay you about $10 a ton if you get it out of there. Right, because they're paying huge amounts of cost.
They're paying to haul it to the dump.
They're paying $50 a ton to get it into a landfill.
What's the particle size of that material?
It's pretty. It's really fine.
Think a cloud.
Yeah, we have a particle size analysis on it, and I forget what the percent is that will pass 100 mesh screen, but a lot of it does.
The question being though is, you talked about test results at 2 different points in the process that you were extracting the fly ash.
Right.
Can you elaborate on that?
Well, okay, so I'm not an engineer, but they use what's called a fluidized bed incinerator when they're burning some of this. They burn part of their waste material to generate heat for their paper production process. And so the fly ash that comes out of that fluidized bed is a prill. Okay, the problem is it's produced at about 500 degrees centigrade. And it's about 60% calcium oxide, but it's about 30% silica oxide. And so what do you get when you heat silica oxide to 500 degrees centigrade? You get glass. So basically you get calcium oxide and a few other things embedded in that glass. And so in our hands, that one treatment, I didn't really say very much about it. When we first started these studies, we didn't know much about that form of the fly ash, so we wanted to see if it was reactive. And basically, it's very weakly reactive, the prilled form. So it comes out earlier in the process. The fine form of the fly ash basically comes out in the evaporation stack So it's very finely textured.
Did some research 5 years ago on the pulp mill down at Pasco, and I was concerned of metals in it.
Right.
I don't know where you're at with this stuff, but I think you could go 1 or 2 times, but I don't know how much you could put on and not get into a heavy metal problem.
Well, that's a great question, and that's one of the issues of concern. So we're getting our material analyzed for heavy metals, and my understanding, I've talked with Department of Ecology and some other people about this, and mostly because their material is all originating from plant products, heavy metal issues are much less of a concern, lower concentration of heavy metals than if you were dealing with something like coal fly ash. So, but I don't have the numbers, but that's something that we've definitely been talking about. Thanks.